All right, guys, you will be designing an experiment that is going to test the impact of some environmental factor, something of your choosing, inquiry-based, once again, do something relevant, on the reaction rate of an enzyme-mediated reaction. All right, so let me introduce, first of all, the stuff that's up on the board, and then I'm going to go through the demo of the titration for you guys. All right, so going over to the board here. Okay, so the reaction that we're going to be working on the decomposition of is hydrogen peroxide. And hydrogen peroxide is going to be able to be decomposed into water and oxygen. It happens spontaneously on its own, very slowly, but with the help of the enzyme catalase, that reaction occurs real quickly. So that is the overall reaction that we are going to be working on testing how fast it happens in various different environmental conditions with the presence of catalase. All right, so we're going to be doing, in order to make this work, we're going to be doing a titration, and the titration is going to utilize, here's your hydrogen peroxide, potassium permanganate, which is purple in color. By the way, notice I'm wearing my lab coat and I will be wearing my goggles because potassium permanganate, first of all, stains. So on Thursday when you're doing your lab, you want to make sure that you don't wear your best white clothing. And then we also will be working with sulfuric acid. Sulfuric acid, of course, is an acid. That's kind of dangerous. Definitely wearing goggles for this. So those are the reactants in this titration reaction. Okay, so that's different than the overall reaction that we want to test. It's the titration reaction. And then the products here end up being colorless. So your reactants are purple, but your, well, the potassium permanganate is purple, but the overall products of this are colorless. And so what you're going to end up getting when you do this reaction is you're going to end up getting clear material as a product as long as you have enough hydrogen peroxide around. Hydrogen peroxide is the limiting reagent, so once you run out of hydrogen peroxide, then the potassium permanganate is in excess, and the product then, because you still have this around, will end up being purple. All right, so what ends up happening then is the amount of potassium permanganate that you titrate in is going to end up being proportionate to the amount of hydrogen peroxide that's in the solution in the first place. And so we're going to be using that as an approximation to tell us how much hydrogen peroxide is present. Okay, so the materials that you're going to be needing to do this lab. Okay, so we got the materials set up over here. We have a burette. It's a graduated thing. It's got some potassium permanganate in it, so you notice the purple color. It has down here, it has a stopcock on it that you will be turning carefully in order to titrate this through this pipette tip down into your solution. You also have some sulfuric acid, and you have hydrogen peroxide in little cups, and then there are also these syringes that are labeled so that you don't cross-contaminate. Okay, so here's how you're going to do your basic titration. All right, so we're going to start out with putting on our goggles. Definitely have to wear eye protection. All right, and we're going to find out how much hydrogen peroxide there is in the hydrogen peroxide solution to begin with, okay, without the enzyme present at all. So in order to know how much was broken down, you have to know how much you started with, and then you'll have to also know how much you ended with and figure out from subtraction then, therefore, how much was broken down. So I'm going to take with my syringe, that is my hydrogen peroxide syringe, I'm going to very carefully measure out 10 milliliters. Milliliters are the same as cc's, but I think these are labeled milliliters anyway. And you're going to put that into a beaker. Okay, so that part's done. All right, then this would be the step where you would be adding the catalase, the enzyme, if you were going to be doing an enzyme trial, but this is just the baseline trial. I don't want to digest any of this. I just want to know how much hydrogen peroxide, how many hydrogen peroxide molecules there are in this, because it's not pure hydrogen peroxide, there's also water molecules. And so we have to know how many hydrogen peroxide molecules there are in this concentration. All right, so because we need to keep everything the same as when we end up having our catalase present, we would be adding 1 milliliter of catalase to our treated ones, but this one being non-treated, we have to add 1 milliliter of water. Okay, so you're going to use your graduated pipette at the tip of the stem. There's a line on there. That is 1 milliliter, and you're going to add that. Okay, so that's just in order to make the concentration the same. All right, now if this was the enzyme-mediated reaction, you would have the stopwatch going because you need to also keep time constant. So I'm going to swirl and I'm going to be timing for, nothing's happening here, but I have to be timing for, let's say 30 seconds. It doesn't matter how much time you use as long as it is consistent. So I'm going to let this go, 30 seconds, tick, tock, tick, tock, tick, tock. Okay, after 30 seconds is up, then we need to stop this reaction, and you need to be ready to go with this. So during that 30 seconds, or somebody else in the lab group needs to be in charge of the sulfuric acid. And you will have 10 milliliters of sulfuric acid all measured out and ready to go, maybe before you start your reaction, definitely by that 30-second time period. And then you're going to add your 10 milliliters of sulfuric acid. Okay? All right. So swirl that a little bit around to mix that up. The sulfuric acid, the point of that, besides the fact that it is one of the reactants, but it also stops the reaction. And the reason it stops the reaction is because the sulfuric acid, being so acidic, denatures the enzyme. Okay, so the protein itself ends up being misfolded, and now its active site doesn't bind to the substrate anymore, and so the reaction just stops. Okay, so we've got some liquid in here now. It's actually 21 milliliters. We're going to do a step that is called the transfer step. This is actually more than we need to do for our titration. We're going to kind of reduce the amount of potassium permanganate that we use by only using 10 milliliters of this at a time. Plus, that also gives you a second shot. So if you screw up the first titration, you'll still have 11 milliliters left so that you can do a second one. So I'm going to use my graduated cylinder to very carefully measure out 10 milliliters of this mixture. Okay, and all the measurements here, this is a pretty picky lab. Everything really does need to be precisely measured in order to get results that are correct, appropriate. Okay, so now that I've transferred, I have 10 milliliters. I'm going to bring my burette here into view. So now I'm going to titrate into there, and as I titrate, there's going to be a reaction that's going on because now I'm adding the final reactant that we need in order to get the titration reaction to go from the board. So it's going to be purple going in, but as we swirl it, it'll fizz and stuff like that. The results of the product should be colorless until we run out of hydrogen peroxide. All right, so titrations. One of the things you want to try to avoid doing is going past the end point, which is, you want to get to a point where you're just the very last drop, that one more drop added is the final drop that was needed in order to make it stay the pink color. Now this one is going to take a little bit because we do have pretty much hydrogen peroxide in our solution. This was our starting our baseline, so it's going to have as much hydrogen peroxide as is possible there. None of it has been broken down by enzymes. I did measure this beforehand, you guys. You should have measured also on your burette beforehand because you need to know how much potassium permanganate you're putting in. It was at 9.2. So you needed to measure that before you start your titrations. Going a little bit more slowly now because it's taken a little bit more time for that to clear. Based upon previous students, one of the things you want to be careful for is sometimes you end up with the, we're very close to the end point now. Sometimes you end up with a little bit of the potassium permanganate held up in the foam. So do make sure that you are mixing very well. This is really close to the end point here. So if I can, see if I, now on film here, if I overshoot the end point, everybody will know it. All right, not too bad. I put in 2 drops. I kind of only wanted to do 1. But now I've got a pink liquid. All right, so I am past the point where I have any more hydrogen peroxide. There's not any more left in my solution. So I'm going to want to measure on my burette. I was at 9.2. Now I am at, I was at, looking upside down, the readings are upside down. I was at 8.8. So, because the reading's upside down. I was at 8.8. Now I am at 11.6. Okay, so 8.8 minus 11.6, that would be the amount of hydrogen peroxide that is in the solution. Okay, so you go through and you'd add that. I don't have my calculator ready to go with that. Hang on. I'm back. Okay, so taking a look at my calculator here, we have 11.6, and we started with 8.8 in our burette, so that means we used 2.8, and I'm going to just use one of these table markers here, and I'm going to record that we used 2.8 milliliters. Okay, I wrote it on the table. I'll show you when we're done. Okay, so that's my first reaction done. I know my baseline. All right, now I need to repeat this, make sure everything is rinsed out, everything is dried out. But this time I'm going to use the enzyme, and so we're going to see how much the enzyme breaks down in 30 seconds. So I need a new 10 milliliters of hydrogen peroxide, measured out well. Okay. All right, now I'm going to get my sulfuric acid ready to go because I am here by myself, so I'm going to be the one that needs to make sure that I hit this right at the end time because my catalase is going to break down a lot of this. And I honestly didn't, I just mixed up some catalase real quickly. It's probably pretty concentrated, so we're probably going to break down a lot of the hydrogen peroxide that's in here. The catalase, by the way, is from beef liver, but it is in really pretty much any living cells. Hydrogen peroxide is a byproduct of metabolism. It's something that is toxic, and so we have this enzyme in our cells. Supposedly you're supposed to be able to extract it from turnips and stuff like that. I tried that, didn't have very good luck, but there are recipes for being able to do that out there. Get them from yeast, I guess you're supposed to be able to, but the beef liver works pretty well. Okay, so I gotta make sure I'm ready with my timer, and go. Okay, so now with the catalase in here, it's going to be fizzing because it's breaking apart the hydrogen peroxide and it's giving off oxygen bubbles. Okay, so in the bubbles there's oxygen, and we're at about 15 seconds here. You can see fizzing, 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 fizzing. That's as the hydrogen peroxide gets broken down. 5, 4, 3, 2, 1. Stop the reaction with the sulfuric acid. Give it a little bit of a swirl, make sure we're stopped. Okay, now I'm on my transfer step. Okay, got my graduated cylinder. I want to transfer 10 milliliters of this to a new reaction container. Okay, 10 milliliters with my graduated cylinder, measured very carefully. Use the meniscus. Okay, save that for later in case I screw up. Okay, now what do you think? Do you think that this reaction is going to go more quickly this time? Do you think I'm going to reach my end point by letting more in or by letting less in? Okay, is it going to go faster or is it going to go slower this time? What do you think? Think about it and we'll see what ends up happening. All right, so once again, you should always measure where you're starting. Now I'm starting where I ended, so I'm at 11.6. All right, ready to go here with my 10 milliliters that has been transferred. And I'm going to be careful this time and see where we're at. I was careful last time too. That was actually pretty good titration. Okay. Let's see if I can get it to go drop, drop, drop. There we go. Swirling. Whoa, stop. Some of it was getting hung up in the foam on the top, and I wanted to make sure that that didn't end up not being mixed in and just that little bit shooting us past the end point. Ooh, getting closer now. I can't see. was lucky that time. A little more came out than what I wanted. Okay, now it's dropping. Whoa, stop. This is a little easier when you have two people. You can have one person swirling, have the other person running the stopcock. Oh, there you go. I did overshoot that one a little bit. Probably should have used a little bit less than what I did use. But there we go. There's my end point. If I was really being exact, I could go back and double check it with my leftover. All right, got to make a reading here. I am at 15.3. All right, so 15.3 minus, we had 11.6, used 3.7. That is weird. I used pretty much that time, so I don't know. Something is a little bit funky with what I did there, but nevertheless, that is how you would end up doing this titration. So if I had, I should have had more than I should have used less when I had the enzyme present because the enzyme broke down some of the hydrogen peroxide. But that is not what I ended up seeing. So if this is my lab, I would go back and redo some of that. So of course, in my demo video, of course, that ends up being screwed up. But it is a very touchy reaction, and it's something that you need to make sure that you're very careful with your measurements and make sure that that's all good. But that's generally how you do this. You're probably going to be testing something like, well, I'm gonna let you brainstorm on that. I can help you out in class if you need help on that in class. All right, good luck.